Can watch this later. There we go. Okay. So does anyone have any questions in particular that you want to go over or topics? Anything you want to practice with, like chemistry?
Definitely things like the macromolecules,
Okay.
and because I didn't take chemistry in high school. So
Yes. Yeah, we can definitely go over that. Yeah, I think that's a good idea. So, let's go over some of these macromolecules first. And I definitely think that the chemistry portion is hard. Chemistry was not my favorite subject. That's why I ended up in biology, not chemistry. So, um,
Well, I said I feel like college kind of presumes that you took all the high school classes, and that definitely didn't happen for me with chemistry.
yeah, I know. Yeah, Tristan, it's like some people took chemistry in high school, but a lot of people didn't. So, yeah. And so they kind of assume, yeah, everybody's had this. And it's like, no, they haven't. So, yeah. So we'll go ahead, go through the organic molecules, the macromolecules, and make sure that people have got those down. Um, so let me just pull up my whiteboard because I like to draw things. So let me find it. Mhm.
You little puppy, little puppy, lay down.
Okay, hold on. It's like they've changed things around on here, and so I'm still trying to figure out like where stuff is. Not that. It's like I was just on here last week, and it seems like they changed things from last week. Like, why did they do that? Okay, I must be on the wrong thing. Oh, they made like a new button. Gotcha. Okay. Me and technology don't get along. Okay. All right. So let's start with our first macromolecule or organic molecule. Um, and we've got, whoops, four of them that we're going to be going through here. Okay, and with the name macromolecules, what's macro referring to?
The bigger molecules.
Yeah, so these are going to be big molecules, and these are all going to also be organic compounds or organic molecules as well. So, how do you recognize that it's an organic molecule? What's it made out of?
Yes, carbon. Carbon.
Yeah, it's always going to be carbon based. And the other thing that always gives it away too is you're always going to have a carbon and a hydrogen in their organic molecule, and then they also will a lot of times have oxygen or nitrogen, and there's other things in there too. But if there's always a carbon and a hydrogen in there, it's got to be organic molecule. So that's like a dead giveaway. So these organic molecules are always going to be carbon based. Okay, so that's another thing to kind of know. Okay, so we've got four of these organic macromolecules. So what should we put on our list here that our organisms are going to be using and that we
So
also,
protein,
yep. So we got proteins. What else?
Carbohydrates,
Yep, carbohydrates,
lipids,
lipids, yes. And what else? We got one more.
nu nu- I know it's the DNA and RNA one. It's
Right.
the
Nucleic acids.
nucleic acid. Yes.
Yeah, some of these words are like, who named these? It's like, they're tongue twisters. Okay, so these are our four organic molecules or macromolecules that all living organisms need to survive. So let's start with our proteins. So I do want you to know what the uses of all of these are, what some of their functions are. So what about for proteins? What are some of their functions? They can actually be used for a lot of different things. They make up your hair, don't they? And your nails. They make up those things. Um, and at least that's, in us and mammals. Um, but we'll talk about later when we talk about cells. We also use them for enzymes. They make up enzymes.
Mm.
And enzymes are going to help speed up the chemical reactions in the cell. Because if we didn't have enzymes, we might be waiting years for a chemical reaction to occur. And we wouldn't be here if we didn't have enzymes. So enzymes are okay. Um, some other things, we actually can use proteins to transport things. So if you think about like our red blood cells, they are filled with a protein called hemoglobin,
Mm.
oxygen attaches to. And the hemoglobin is actually a red color, and that's what makes our blood red. So that's that protein. So it can be used for transport. Um, and there's some structural roles for our proteins as well. So inside of our cells have a little skeleton inside of them. Okay, this little protein skeleton. So they actually call it a cytoskeleton, cyto meaning cell, and then the protein part that makes up the skeleton, um, they just call it the cytoskeleton. So yeah, so there's lots of roles for proteins in the body. They make up our antibodies that we use for defense. Um, so it helps protect us from infections. I mean, pretty much if you can think of, you know, some role in the body, probably a protein's going to do it. So proteins are used for lots of different things. And that's just because they come in lots of different shapes, right? Okay. Um, Okay, the other thing that I would know about these macromolecules, besides their functions, like what are we using them for? Um, also know what's their monomer and what's their polymer called? Okay, so first of all, what does that word monomer mean?
It's the smallest unit that
Yeah,
combines with other units to make larger units.
yeah. So mono meaning one, and mer just means part. So a monomer is just the basic building block or subunit. Okay, and then if we put the monomers together and join them with chemical bonds, then we make the polymer, because poly meaning many. So you put many monomers together to make the polymer. Okay. So with our proteins, what's going to be the monomer? What's going to be our little subunit that we're going to start with to make our protein? Starts with an A.
Amino acids.
Yeah, good, Bushra. So those would be your amino acids. And so if we join a bunch of amino acids together, so if we have all of these amino acids together, eventually we're going to form our polymer, which is our, oops, don't need a P there. Which is our polypeptide chain. Okay, or protein. Okay. All right, and then we also have, with the proteins, we have 4 different levels of folding. Okay, and that'll give the protein the specific shape. And the shape is really important with the proteins because that'll determine its function or its job. As proteins can come in all different shapes. And that's why we have all these different functions, all these different jobs for proteins because they have all these different shapes. Okay, so with our proteins, we've got the primary structure. And then we had secondary, tertiary, and quaternary. Okay, so what would be your primary protein structure? How do we describe that one?
So I'll admit that I wrote down the 4 structures in my notes because I do not understand. It seems
Okay.
like it's English, but it doesn't make sense to me.
Okay. All right, well, let's look at a picture of this because I think this will hopefully help. So let me find my nice picture. Oh, there it is. Okay, so let me just pull this one up and we'll look at a picture. There it is. Okay. So we've got, in this picture on the bottom, we've got our primary, secondary, tertiary, and quaternary protein structure. And these are just the different levels of the protein structure. So when you go to make your protein, we're going to start with the primary structure first. So this is just kind of the first level. So the first level, this primary structure, is just the sequence that the amino acids are put together in. Okay, so it's just this chain of amino acids, just the sequence that they're in. That's the primary structure. That's it. Okay, and then I've written up here, what each one is defined as. Okay. All right, and then you take that chain of amino acids, that primary structure, and now you're going to start folding that more to get your secondary structure. Okay, and the secondary structure you can either fold into a coil, and that would be called an alpha helix, or you can fold it into kind of like an accordion structure, and that's called a beta pleated sheet. Okay, so that would be a secondary structure. Okay, so either these alpha helices or beta pleated sheets. And then the tertiary structure, all that is it's just the overall 3D shape of that polypeptide chain. Okay, because you've now taken that secondary structure and then folded it some more, and now what's just that resulting 3D shape of that polypeptide chain? Okay, and so if you just look at the overall 3D structure, is it kind of a rounded shape? Or maybe it's more like kind of a rope-like shape. Okay, just whatever its three-dimensional shape is. Okay, that's the tertiary structure. And that's all it is. Okay, and then the quaternary structure, you're only going to go up to this quaternary level if you've got two or more polypeptide chains that are together. It's like you can see in this picture, you've got a light gray and a dark gray polypeptide chain that are together. So you've got two polypeptide chains there. So that would give you quaternary structure. But if you only had one polypeptide chain, you would just stop at tertiary structure and you'd be done. There wouldn't be any quaternary structure. But if you do have two or more polypeptide chains together, then you would have quaternary structure. And that's all it is. Does that make sense? Is that good, Bushra and everyone else?
That actually makes sense. I didn't realize that it was like a sequence, like you had to have the first structure to get to the second, to the
Yes.
secondary and tertiary. I just thought that those were like four different kinds existing
No,
separately, but that didn't make sense.
yeah, you got to start with the primary, and then from the primary you fold it more to get the secondary, and then the secondary you fold more to get the tertiary, and then from the tertiary you'd have to add another, at least one more polypeptide chain or maybe more to get the quaternary structure. Yeah. Okay, so that's all you need to know for proteins. Does everybody feel okay with proteins? So you just need to know kind of, and remember, these are all going to be multiple choice or matching. So if you know, are okay with, you don't have to write any of this out, so that's good. And you've got your note sheet that you can have front and back. So as long as you're okay with, the basics here, with what we just talked about, knowing, what proteins are used for, what the monomer is, amino acids, and the polymer would be the polypeptide, and then just the four levels, primary, secondary, tertiary, and quaternary. That's all you need to know for proteins. So
For the note sheet, is it just like a normal piece of paper?
yeah, just like
Okay.
regular eight and a half by 11 sheet of paper. You
Okay.
can have, yep. So,
No
okay.
poster boards.
No, Tristan, no. Board. Nice try, but not a poster board. So, okay. So let me see. Oops. Not that one. Oh, back to the one we were at. Okay. Okay, so that's proteins. Next, let's take a look at our carbohydrates. Okay, so I'm just going to erase what we had here, and then we'll go to our carbohydrates next. Okay, so our carbohydrates. What are we going to be using carbohydrates for?
Energy.
Yeah, they're definitely an energy source. So we'll break them down and use them for energy. And anything else? What about plants?
I don't remember.
What about wood? What's wood made out of? And paper.
Do they, excuse me if I'm mixing things, but do they form the cell wall?
Yeah, they do. So with plants, they make their cell walls out of cellulose. Okay, and that's actually a carbohydrate. So they can use these carbohydrates for structure, okay, to make their cell wall out of that. And that helps give their cells some shape and then also helps support the plant, gives them some rigidity, and actually helps make the wood if you're a tree, things like that. So yeah, so it can be used for structure in some organisms. But our cells, we don't have cell walls, so yeah, we don't do that part. But okay. So the carbohydrates. What would be our monomer, our building block, our subunit?
Right?
What?
Sorry, monosaccharide? It's
Sorry. Yeah, monosaccharide. Right, and that one's easy to remember because mono means one, right? And saccharide, if you think of like the artificial sweetener like saccharin, that reminds you of sugar, right? So literally means one sugar. Okay. And so if you put those together, join them together with those covalent bonds, they will form the polymer. So what would our polymer be called?
polysaccharide.
Yeah, it would be your polysaccharide. Okay, and poly just meaning many, so you've got many sugars joined together. So for example, like in us, a monosaccharide could be like glucose. And then for us, like if we've just eaten, and we've got lots of glucose running around and we haven't, we've broken enough down for energy and we're like, oh, we've got some extra glucose. What we'll do is we'll save the extra glucose for later and we will store it as the polysaccharide. We'll hook a bunch of glucoses together and make the polysaccharide glycogen. And we'll store that for later because when we evolved, we never knew when our next meal was coming because we didn't have grocery stores. So that's why we do that. Okay. All right. And then another term to know, what's a disaccharide? We're kind of in between here.
Two monosaccharides combined?
So di meaning two. So you've got two monosaccharides joined together, and that would be a disaccharide. Okay, so like the sugar in milk, lactose, that's a disaccharide, for example. Right. Good. Okay. So that's what you need to know about the carbohydrates. So any questions about the carbohydrates? No? Okay.
Sorry, just one question. Do we need to know all the examples of monosaccharide, poly and disaccharides? What like is there?
No, you won't be asked like, oh, is this a monosaccharide or a disaccharide?
Right.
You know,
Okay.
like, oh, you've got fructose, and is that a mono or a disaccharide? Yeah. No, we're not going to ask you that. Yeah. But just knowing the terminology, like one sugar versus two sugars versus many sugars, and just knowing like what the monomer and the polymer is. And that's good. Yeah. Okay. So everybody feel pretty good about the carbohydrates?
Yeah.
Okay. All right. And then we'll move on to our lipids. Okay, so what are the functions of the lipids? So think about us, like what do we use lipids for?
Isn't it like stuff in our blood? Or because like
There
I
are
don't know.
some.
I think it's stuff in our because like when I went to the doctor, I got like a lipid panel where they drew my blood.
Mhm.
It's something like that that tells you like the glucose and
Yeah, so they
a
tell you
bunch of
like
other
your
stuff about your blood.
cholesterol levels, your triglyceride levels. That's what's in a lipid panel, right? And that helps tell you about like if you have high cholesterol and maybe you need to do something about that with your diet and exercise and stuff. Um, yeah, but what do we use these lipids for in our body?
I will admit I don't remember their function in the human body, but I do remember the image of that otter and
Ah,
how like the its skin has it so that it protects it from the elements. So it's like it provides an
ah,
insulation.
right. Yeah. So, and we use lipids for insulation too, right? Okay, because our fat stores in us do provide insulation and keep us warm in the winter as well.
Mm.
Okay, and then it can also provide a cushion, right? We do have fat pads. Um, like on our rear end, we're all sitting on right now. There's fat there. There's some fat on the balls of our feet too. And around certain organs, like around your kidneys, there's some fat helping protect some of these organs. So that does provide a cushion. And then we also use these lipids as an energy source too. Okay, so if you haven't eaten for a while, you will go down and start breaking down your fat stores in your body to provide energy for your cells.
It's funny you say that about the cushion because I lost 70 pounds in the last year and I'm now having problems with my tailbone because I have like no cushion
Yeah,
now.
that is true, Tristan.
Yeah, I
Yes.
have to sit on a donut pillow all day.
Oh no. Oh gosh.
Yeah.
Well, congratulations on losing the weight.
Thanks.
I'm sorry you have to use a donut.
Yeah. I was like thinking I was gonna have a hot girl summer and then I had to carry a donut pillow around all summer.
Oh no. Well, that's not so attractive, but oh, always trade-offs.
Yeah.
Okay. So there's really no one monomer or polymer for lipids because they're a pretty diverse group. And the reason we put all of the lipids together is because are they all going to be hydrophilic or hydrophobic?
Ooh, hydrophobic.
Yeah, they're all hydrophobic. And that's why we group all of the lipids together. Bless you.
Oh, sorry, I thought it was on mute. Excuse me.
Oh, you're fine. Okay, so I remember that. That's why the lipids are all together because they're hydrophobic. They don't like to mix with water. So if you remember, like with salad dressing, if you pour some salad dressing or olive oil or something into some water, it will not mix. It will huddle together. It won't mix together with the water. So they're hydrophobic. Um, so the lipids we talked about, we had triglycerides, and these are better known as the fats. Okay. And we talked about saturated fats and unsaturated fats. And which ones are better to eat in your diet?
Unsaturated.
Unsaturated?
Yeah, good, Ivy. Yeah, it's going to be the unsaturated fats are healthier. Okay, and these will be coming from your vegetable sources. So like avocados, vegetable oil, olive oil, those kinds of things, canola oil. Whereas saturated fats are more unhealthy because they're gonna be coming from animal sources. So like on meat, butter, things like that. They're gonna have the saturated fats. And the saturated fats you don't want to eat as much of because they're more likely to raise the bad cholesterol. Okay. And that can lead to heart attacks and strokes later on in life. So we don't want to have too high of cholesterol. Okay. And then we also had our steroids. And the steroids are all made from cholesterol. Okay. And these steroids, we use cholesterol in our cell membranes. So it is part of the cell membrane. And then we also make from cholesterol, we make all of our steroid hormones. So like all sex hormones like estrogen, testosterone, progesterone, those are all made from cholesterol. Okay, so those are examples of steroids as well. Okay. And then the other lipid we have are phospholipids. And these make up the cell membrane. So these are pretty important. It's well.
And these are all like examples of lipids.
Yes. Yes. So there's three types of lipids. I mean, there's more, but the triglycerides, steroids, and phospholipids, these are all types of lipids. They all have very different structures from each other. So they're all made differently, different parts. So there's not going to be one particular monomer or polymer for the lipids. But we just group them all together because they're all hydrophobic. Okay. All right, everybody good on lipids? Okay.
Wait, what about the waxes? Isn't that a lipid?
Oh, yeah, the waxes are, but these other three are more important.
Oh, okay.
But yeah, waxes are a lipid too. And on us, the only wax we have is the earwax. So yeah.
Interesting.
Um, but yeah, but others, there's wax in other, species, other places. They use it for different things, but usually it's to help with waterproofing. Yeah, to make kind of a impermeable layer. So, but yeah, but we've got earwax. So
Boy, we got the short end of the stick there.
I know, but our skin's pretty waterproof. So
Yeah, that's true. That's true.
okay. So our last organic molecule is our nucleic acids. Okay. So what's the function of our nucleic acid?
They contain information, like
Yeah.
genetic information.
Now we're going to store our genetic information. So that'll be our DNA doing that. And then they're also going to help make proteins. And the RNA will do that. So that's why we've got the two forms. Okay, and let's take a look at our monomers and our polymer. Okay, so what's going to be our monomer, our subunit for nucleic acids?
Is it a nucleotide?
Yeah, good, Tristan. It's a nucleotide. Very good. Okay. And then if we join a bunch of nucleotides together, we can make our polymer, which is just our nucleic acid, which will be either DNA or RNA. Okay. All right. Just some differences between the DNA and the RNA. Obviously, they have different functions, which we talked about. But remember that DNA is going to have double-stranded. So it makes that double helix. And RNA is just going to have a single strand. So it'll be a big difference between the two. Okay, and we'll talk more about some more differences between DNA and RNA later on when we get to the genetics. So, okay, so those are all of the macromolecules or organic molecules. Everybody feel pretty okay with those? Yeah.
Yeah. Can we just talk really quick about the difference or what transcription and translation is,
Oh, yes.
please?
Yeah. Mm-hmm. Okay. So in your DNA, you've got genes, which will code for proteins. Um, and when you want to take that gene and say, hey, maybe we need to make some insulin protein hormone, because we just ate a big meal, your cell would say, okay, we need to transcribe that insulin gene. Okay, so the first step in making the insulin protein hormone will be to take that insulin gene in the DNA and you're going to transcribe it. So the first step is to do transcription. And the way I remember this is transcribe means copy. So what you're doing is you're just making a copy of the DNA. And that copy you make is messenger RNA. Okay. So we're going to transcribe the insulin gene, that DNA first. And that copy we make is messenger RNA, the mRNA. And then once we've got that copy, that messenger RNA, we're going to read the code. We're going to translate it. So step 2 would be translation. So we're going to read the code on the messenger RNA and figure out which amino acid we need to put in which order to make our protein. Okay, and then we've made our insulin. So the way I keep this straight, how I know, because they both start with trans, and it's really easy to get them confused, is I just remember that when I look at these two words, the C comes before the L in the alphabet, so I know that transcription always comes before translation. I have to use little tricks to get my memory to work. So I
Me
make,
too.
yeah, it's the only way you remember stuff. So,
That's a good one, Kate. Thank you.
okay. Any other questions about any of these macromolecules? Everybody's feeling okay? Okay, should we do any chemistry practice problems with
Yes.
deciding Ivy says yeah, definitely yes. Okay. Um,
Yes.
okay. All right, River wants to as well. Okay. Let's say that I give you um let's see, how about I give you this atom? Let me see. How about we do okay. How about I give you this? I'll give you potassium. And let's say the mass number is 39 and the atomic number is 19. Okay, what's our number of protons?
19?
Yes, it is 19. And how we know that is the atomic number is always the number of protons. Okay, how many neutrons is there?
Winnie?
There are 20. Good, Bushra, right. So the mass number is the number of protons plus the number of neutrons. So all you have to do is just subtract these two numbers, and that'll just give you the number of neutrons. Okay, how many electrons are there?
19.
There's also 19. So as long as you say that this is not an ion, it's electrically neutral, the number of protons is always equal to the number of electrons. Okay. All right. Should we draw a Bohr model for this? Okay. So we know we have 19 electrons. So how many electrons go in the first shell?
Two.
Yes, the maximum number of electrons you can put in the first shell is always 2. Okay, what about our second shell? How many electrons can we put in the second shell?
8.
8. Okay, so we're up to 10. How many electrons can we put in the third shell?
9, but it won't be stable. Oh no, wait. Sorry, it will also be 8.
Okay. Okay, and that means in our fourth shell, we've only got one electron left, right? So that would be our Bohr model for potassium. Okay, so just remember that the first shell is always 2, and any shell after that, for our purposes in this class, 8 would be the maximum number in any other shell after the first shell. Okay, so you go around and fill up 8, and then whatever you've got left over would be in the last shell.
Mm.
All right, and would this react with another atom? Does it have a full outer shell? So it would react, wouldn't it? Because it doesn't have 8 in the outer shell. So it would react. Okay. So yes, it would form a bond with somebody else. Okay. So if they have a full outer shell, like if they only have one shell, this first shell, and they've got 2 in there, it would not react, wouldn't form a bond. If you've got, you know, an outer shell, maybe the second or third one has 8, it wouldn't react, it wouldn't form a bond. Okay.
So all the little dots are like signify electrons, right?
Are electrons, right?
Okay.
Yeah. Yeah. And I didn't draw the neutrons or the protons in here, but I could put, hey, there's 19 protons and there's 20 neutrons in the nucleus there. But I just didn't draw them in there. Because usually the Bohr model, they're more concerned about the electrons and what they're doing. So, yeah. Okay. What if we have an isotope? So what if I've got, I don't know, whatever this atom is, I'll just say atom A. We won't say it's anything. But what if I've got, I don't know, let's say we've got 30, $32, and I don't know, maybe we've got. So what would be the difference in the number of neutrons between each of these? And knowing that these are the mass numbers up here. So what is an isotope anyway?
I know that there's something that's not equal in there, but I can't remember if it's electron and protons or protons and neutrons, that the number is not equal.
Okay, so with isotopes, okay, they're all the same element, except there's a different number of neutrons. Okay, so like their atomic number would all be the same. Okay, but you can see with this mass number being different, you haven't changed your atomic number on the bottom here. It stayed the same, so the number of protons is always the same. But your mass number has changed, isn't it? Okay, so your number of protons is the same, but you've got a different number of neutrons, right? So you can see that each one is, we've increased by 1 neutron for each one. Okay. And how you tell if you're looking at a mass number or an atomic number, the mass number is always going to be bigger, isn't it, than the atomic number? Okay, because your mass number is the protons plus neutrons, right? Whereas your atomic number is only the number of protons.
So are protons and electrons always the same number?
Yes, as long as it's not an ion. But if it's an ion, they're not equal. That's what makes it an ion. Right, so we'll do that example next. Okay.
Sorry, I don't mean to be rude, but I
Yeah.
have childcare only till 4:30, so I need to drop
Oh,
off. I'll watch
yes.
the recording after that.
Yeah. Thank you for
Thank you
coming,
so
Bashar.
much. Thank you.
Okay, bye.
Bye.
Okay, so let's do an example with an ion next. Okay, so let's say that we've got whatever element this is, A. And let's say that we're given this information that we've got, let's say we've got atomic number. Say it's 20. And let's say that our number of electrons is 21. Okay, so let's see if there's a charge on this atom. So is this an ion or not? What do you think? The first thing I would do is let's figure out the number of protons, right? So how many protons do we have?
20.
Yeah, good, Tristan, we have 20. Okay, so a good way to do this is, remember, protons have a positive charge and electrons have a negative charge, right? So all you're going to do is you're just going to add these two numbers together. But we're just going to put the charges on from the positive from the proton and the negative from our, so if we've got plus 20 from the protons, so we've got 20 protons, and we've got 21 electrons, and we put a negative sign there for the electrons. If we add those together, we get a charge of -1, right? So that would be the charge on our ion. And we would just write it as a minus sign. They don't actually write it as -1, so they just put a minus sign. Okay, so our charge would be -1, right? Okay, now what if we had this one, what if we had, I don't know, whatever this is. And so we had an atomic number of, say, 14. And our number of we have 11 electrons. So what's going to be our charge on this one?
Would it be +3?
Yeah, it'd be a +3 charge. Okay, so our ion would have a +3 charge. Yeah, good. And that's how you figure those ions out. So ions just don't have an equal number of protons and electrons. That's ions. So does that feel better with figuring
A
those out?
little bit. I've always struggled with this stuff.
Yeah, but does it make it easier, how we've done it, you know, putting a plus sign on the protons and then a negative sign on
Yes.
the electrons, and then you just add them together, and then you can figure
Yes.
pairs that way?
Yeah, that makes it a little bit easier.
I
Okay.
think I understand all of it up until like the math equation, and then I'm like, oh, I don't know.
Oh, okay. Um, trying to think what else I can do, but you can see, Tristan, that if you look at these two numbers, that your number of protons and your number of electrons, they're not equal, are they?
Yeah, and when I first looked at that, I thought, oh, that's easy. It's going to be +3 just from those two numbers. But then when you wrote them down here, like,
You can
then
do
I
it
was
that
like,
way too.
oh,
You can do it that way too. You don't have to do the equation. Because if you can just look at that and say, hey, I've got 3 more protons than I do electrons, yeah, that's going to be a +3 charge. That's fine.
okay, okay.
Or like on
That
the,
itself made a little bit more sense.
hey, if that works for you, that's great. There's different ways to do this. So, and then on this top problem, if you want to look at it and say, hey, I've got 20 protons versus 21 electrons, I've got 1 more electron than I do of the protons, so that means I'm going to have a -1 charge.
Okay, that makes sense.
Want to look at that way? That's fine too.
Okay,
Yeah. So whatever clicks in your brain, yeah, use that.
okay.
So yeah,
I'm
because
kind
there's
of like a writing down type of person. So will we be able to kind of write it out on a paper as well,
yeah, you can
or
have you can
no?
have scratch paper with you while you're taking the
Okay.
test. And you
Okay.
can write all over it. Yeah, that's fine. Yeah. Yeah, I'm very much the same way. I've got to write stuff down too. I got to draw pictures and all kinds of stuff. So yeah, yeah, so that's fine to do. Yep. Okay. So you guys feel better about this stuff?
Yeah.
Okay. What about if I give you um some drawings like this?
Yep, I'm lost.
Yep, done.
Okay. Now you don't even know what the question is. Okay. Is this going to be hydrophilic or hydrophobic?
Yeah, I don't know.
Mm-mm.
Okay. What about this one? Okay. So let me give you some helpful things, because if you see pictures like this on the test and they ask you, is this hydrophilic or hydrophobic? You'll get these right. Okay. So to figure out if these bonds are going to be polar or non-polar, because whenever you have these, you know, dash solid lines like that, that means you've got a covalent bond. It just means that they're sharing electrons with each other. But whether they're sharing the electrons equally or not equally, it depends on if you have an electron hog or not. Okay, if somebody is an electron hog, it means they really love electrons and they really like to pull on them. And it's not a fair tug of war game. And so electrons are going to be spending a lot more time around one of the atoms than the others. And then you get these partial charges. And so this will be a polar covalent bond. And it'll be the same thing over here too. Okay. So if you see oxygen or nitrogen, and they're playing tug of war with like carbon or hydrogen, they're always going to win. Okay. So these are going to be hydrophilic. Okay, because you're going to get these partial charges, and water is going to be attracted to that. Okay, because remember, water,
So that's when it absorbs it.
yeah, they'll mix together. Because water is a polar molecule, and it has these partial charges on it, and so they'll all be attracted to each other and they like to mix together. But if you have really weaklings, like carbon and hydrogen, um, they don't pull on the electrons very much. So these are all going to be non-polar, oops, covalent bonds. The same for those. So there's no charges anywhere, and so water's not attracted to it. So these are all going to be hydrophobic. So they're not going to mix with the water. Okay. So if you see oxygen or nitrogen in the molecule, yeah, so it'll be hydrophilic. But if there's just carbon and hydrogen, yeah, it'll be hydrophobic.
That makes
But
sense.
what if you have this? What if you have oxygen? Like an oxygen we're breathing in. What if you have two oxygens playing tug of war with the electrons? They're both
How would that happen?
so this is the oxygen we're breathing in right now. They've formed covalent bonds between each other, so they're joined together and they're sharing their electrons. But they're both the same strength. They're both electron hogs, so they're both pulling on the electrons, but they're both the same strength. So nobody's going to win the tug of war. And so when you have two atoms that are the same joined together, it's always going to be a nonpolar covalent bond. And it'll be hydrophobic. So when you have two that are the same. Okay, they'll always be hydrophobic.
So I'm a bit confused about like the lines and then the H's about how like I'm like how does that relate to how, if it's polar or non-polar? If there's like the
Yeah.
line and then the H.
Yeah, so this solid line here just means that it's a covalent bond. But it doesn't tell you if it's a polar covalent bond or a non-polar covalent bond. So you just have to know that, hey, hydrogen and carbon are both pretty weak. And so they're going to be pretty evenly matched in the tug of war on the um, electrons, because a covalent bond just means that they are sharing the electrons. But when they're both weaklings, they're going to share the electrons equally. Okay, so they're not going to have one, pulling on them more than the other. And so they'll spend equal amounts of time around each atom. So these will all be non-polar covalent bonds. And that just means, hey, they're sharing the electrons equally. Okay, but if you have the oxygen electron hog sharing with hydrogen or carbon, you've still got that straight line there, just saying, hey, it's a covalent bond, but we just know that, oh, oxygen's a big electron hog, carbon and hydrogen are pretty weak. So oxygen is going to pull harder on those electrons that they're sharing, and electrons will spend more time around oxygen than they do around the carbon or the hydrogen. So we will end up with a partial negative charge on the oxygen and partial positive charge on the carbon and partial positive charge on the hydrogen. So these will be polar covalent bonds. So it just means they're sharing them unequally. So that's all that means. Okay, so there will be some questions on the test where there'll be some pictures like this. And just check and see if there's any oxygen or nitrogen around. And if you see those, you know it's going to be hydrophilic. And if you don't see any oxygen or hydrogen around, then yeah, you know it's going to be hydrophobic.
Okay.
Okay. So that'll help with that. And there were some homework or quiz or assignment problems that were like this. So you might want to go back and review those. Okay. All right. Okay. So everybody feel okay with the chemistry?
I mean, I feel a little better.
Okay. Good.
So
Uh,
I have a question.
yes, Abby.
Um, just so when it's hydrophobic, does that mean is it always going to be nonpolar? And if it's hydrophilic, will it always be polar? Are those words like basically the
Yeah.
same in a sense?
Yeah. And the only other thing that we can also add, okay, would be things that are hydrophilic. Would be if they're polar, um, they have polar covalent bonds in them. Or if they're ions. Okay, so maybe you have like a sodium ion or a chloride ion. See how they have a charge on them? That's going to be very attractive to water. Because water has polar covalent bonds, and so water's like, hey, I like those charges, and I'm going to mix with you. Because if you think about when you put salt into water, it dissolves, doesn't it? It mixes really well with the water. Okay. A polar covalent molecule would be sugar. If you put sugar into water, it dissolves in the water. So that's another good way to remember this one too. Okay, things that are hydrophobic. Okay, things that are nonpolar. A good example there would be any of your lipids. Okay, so like if you poured, some, um, whatever kind of cooking oil you use, olive oil, canola oil, whatever, if you pour that into a glass of water, they will not mix. That's hydrophobic. Yeah. Okay. Does that make more sense, Avi?
Yeah, it does. Thank you so much.
Yeah. Okay. And everybody knows um, ionic bonds. We've kind of been going over the nonpolar and polar covalent bonds. Um, Everybody's okay with the ionic bonds. Um, and what those would be, hydrogen bonds. A ionic bond would form between two ions. So if you had a sodium and a chloride ion, those two charges on the ions would attract each other. And they would form an ionic bond between them. Okay, and that's what forms a salt crystal. Okay. What about a hydrogen bond? What would that be? A lot of times we talk about it forming between like water molecules, but it can form between any two polar molecules. And what it is just the attraction between those partial negative and positive charges. And they just draw it as like a dashed line. Because hydrogen bonds are pretty easy to break. So these form between two polar molecules. Yes. All right, I think that's pretty good for all the chemistry stuff. A lot of chemistry, right? Okay. And everybody feels pretty good about the other two modules, modules 1 and 2. Scientific method, you know the steps. Um, trying to see what else. Oh, make sure you know the properties of life to be able to recognize those. Like, what are the things living things do? Um, make sure you know those. And then I'd also know that hierarchy of organization of living things. So it goes from like an atom to like a molecule to organelles and then cells, tissues, organs, organ systems, organism. And you have a population, community, ecosystem, and the biosphere. So just kind of know that order and kind of what those things are representing. Okay, so I would know those. Um, Oh, and then I'd also know the difference between prokaryote and eukaryote cell. So what were the big differences between a prokaryote and a eukaryotic cell?
One is single-celled and the other one is multi-celled or multicellular, right? Or is
Uh,
it? Oh,
there
it's
are,
in both.
yeah, there's something like even more, like if you look under the microscope at them, like there's something um, like if you look at their cell
Isn't
part,
it like one has a cell wall and the other doesn't?
no,
I think
because
so.
there are some eukaryotes that have cell walls. Something about their cell parts that's pretty big.
One has a nucleus?
Yes, which one has a nucleus?
No clue.
The eukaryote?
Yeah, the eukaryotic cells have a nucleus, and the prokaryotes do not have a nucleus. And the other big difference is the eukaryotic cells have organelles, and the prokaryotes do not have organelles. Yeah, so that's another big difference there. Okay. And then lastly, what are the three domains of life? These are the three big groups of organisms of life.
Bac Bacteria,
Huh?
archaea,
Yep.
and then the one that starts with the E, the U.
Yeah, eukarya, which we're part of.
Yeah.
Very good. Yep. Yeah, so I'd know those as well. Yeah, very good. Okay. So any other questions? I know we're like way over time, but hopefully this has been helpful. Any other questions? Anything else you want to go over?
Just real quick, when you said the properties of life, is that the things that set apart like living things and non-living? The
Yes.
characteristics?
So the things that all living organisms do, so like they have order, they reproduce, they have adaptations, they evolve, they
Okay.
grow and develop, so all those things.
Okay.
Um, so that's how you know that they're alive. Um, so yeah, so I'd be able to recognize those. So yeah, I would know those too.
Do we need to know all the properties of water?
Um, I would just like be aware of them. Um, because water is pretty important for life and just be aware of them, but like they're not going to be like, explain, this property or for that property, but just be aware of that.
Okay.
That's all I'll say.
Okay.
That's not an in-depth thing. So, yeah. Okay, any other questions? And you can always, if you think of something later, you can always email me too, or happen to meet with you on Zoom again too. So either way.
This was really helpful. Thank you.
You're welcome. Yeah, I'm glad you guys all came. So thank you for coming. And
Yeah,
I
this
wish
was very, very helpful.
good, River. I'm so glad. So yeah, and best of luck to you guys on the exam. And yeah, so hopefully everybody gets an A. So
Thank you.
you're welcome. All right, you guys have a good evening.
You
A
too.
quick question.
Oh, yes, go ahead.
Yeah, will we be able to watch the recording today or?
Yeah, it usually takes about a half an hour for them to send me the link to the recording, and as soon as I get the link, I'll put it up in the announcements.
Oh, okay. Thanks so much.
You're welcome. All right, bye.
Bye.